So thank you so much for a nice introduction, Amy. Good morning. And the title of my talk today is Exploring the Diagnostic and Therapeutic Potential of Extrachromosomal Circular DNA in Cancer. The talk is divided into two parts. In the first part, I would like to talk about a robust algorithm which we have designed for the analysis of extrachromosomal circular DNA using whole genome long-read sequencing data. In the later part of my talk, I would talk about microDNAs, which are the smaller form of these eccDNA, and we would discuss that how we are thinking that they could be used as a non-invasive biomarker for early stage breast cancer detection. So let's proceed with the long circle finder story. These eccDNAs are present both in normal and cancerous human tissue. The longer form of eccDNA is known to harbor the cancer gene and attribute aggressive properties to the cancer cells. The cancer patients who have these eccDNA show poor survival outcome as compared to the patients who have no focal amplification or chromosomal amplification. These longer eccDNAs are believed to be caused by shattering of chromosome, the process known as chromothripsis, and religating of the fragments forming circular formations. The eccDNA which have the cancer gene would be selected by the tumor cells, and that would result in the rapid amplification of oncogene through missegregation of eccDNA in the cells. The fragile nature, the variability in the eccDNA fragments, and the extracellular presence makes the sequencing of these entities very difficult task. We are taking benefit of long-read sequencing, which provides an advantage of sequencing the amplicons, especially at the junction sites where the ECC fragments are ligating. I would like to thank Dr. Yoshiyuki Shibata for providing this figure. Here you could see that these are the MYC eccDNA present in the colo 320 DM cell line in this metaphase fish figure. Coming to the workflow of long circle finder, we would start with mapping the long read sequencing data to the reference genome, following the identification of overamplified regions, which would be the regions which are having more coverage as compared to the background. Then in the next step, we would construct a graph for these amplified regions, connecting them together by depth first search. In the following process, we would search for the closed loop in the graph, which would be the eccDNA candidates. The innovative part of this long circle find algorithm is that after the cycle is detected at the ECC junctions, we are taking the reads, supplementary alignment reads, which are spanning the ECC junctions and performing the partial order alignment, thus doing the local de novo assembly at the ECC junction, which provide the base pair resolved refined connections, which can be then subjected to PCR sequencing and Sanger sequencing for validations. So coming to the methodology, in the first part of methodology, we are benchmarking the long circle finder on the simulated data. We simulated 100 eccDNA events at three coverages, 50x, 100x, and 250x, with the background coverage of 10x. The fragment details which are making those eccDNAs are from 3 to 22. We avoided the centromeric region and chromosome Y for selection of the simulated data. For the benchmarking on the real world data, we downloaded the publicly available sequencing data for two cell lines, Colo 320 DM and CHIP212 for running long circle finder, and then comparing it for the other tools which could provide the eccDNA detection named as Coral and Decoy. So coming to the results section, this is a heat map which is showing the F1 score. F1 score is the harmonic mean of sensitivity and precision. On the y-axis, we could see that there are three algorithms details: long circle finder, coral, and decoil for the coverages 250x, 100x, and 50x. On the x-axis, we could see that there are 5 submitted columns for the breakpoint started from 3 to 22. And we could clearly see that for circle detection, long circle finder is outperforming its competitive algorithms, especially at the low coverages of 50x. So the purpose of these algorithms is basically to detect the circles, but sometimes when the data is shallow, the circle detection is not the optimum option. So we go for the detection of the amplified fragments. And here in this heat map, the F1 score for the amplified fragment detection is given. On the y-axis, the three algorithms for three coverages, 250x, 100x, and 50x is given. On the x-axis, the breakpoints are given, and we could see that for the amplified fragment detection, long circle finder is especially outperforming its competitors, especially for the longer circles. The interesting part is this, that we could also see that decoil is also comparably performing better when it is coming to the identification of the ECC DNA fragments, which are the amplified fragments in the smaller circles. Next comes the benchmarking on the real world data. For this, we identified these junctions which are previously reported by short read analysis by Amplicon Architect, and we could see that the ECC junctions which are reported in the literature, we could also find and validate using the long circle finder. I downloaded the publicly available data, and we could see that 6 known junctions which were detected by Amplicon Architect are also detected by LCF, and we validated them by PCR sequencing, for PCR amplification and Sanger sequencing. I would like to thank Dr. Sabah Tufail for doing these PCR amplification and sequencing. This in the figure A, we are showing the graph having the 4 amplicons in the chip 212 cell lines, which are reported in the literature as well. And in the figure B, this is a representative ECC DNA in which the fragments are joining each other with the MYC and gene present at this chromosome 2 fragment. So in this second cell line, Colo 320 DM, we are comparing the output from Amplicon Architect, Coral, and Long Circle Finder. And we could see that we are identifying and resolving the ECC junctions to the base pair resolution, and also correcting the previously identifying junctions, which were away from 332 base difference and 232 base difference. And we could see that we could pinpoint to the base pair resolution in the other 2 junctions as well. So here in this case as well, we had downloaded the publicly available data, and then we could see that the LCF as compared to the Coral is identifying the new junctions, which were misinterpreted by the Coral, and the previously known recurrent junctions as well, which are absent in the ECC DNA negative cell lines. So in this graph, of Colo 320 DM cell line, we are showing the amplicons, and there can be different combinations of ACC DNAs which can be formed. And in the figure B, we are showing the ACC DNA fragments from five of the ACC DNA fragments from 4 chromosome with the MYC gene at chromosome 8. So coming to the discussion, we are in the process of benchmarking using the CHIP212 Colo 320 DM deep sequence data, which is recently been available. In the validation phase, we are doing the PCR detection and of ECC junctions, and we think that we could use this for personalized marker for monitoring the minimal residual disease or early occurrence in cancer patients. Then in the following step, we think that we can target these junctions by sgRNA-based approach. We think that our algorithm LCF is performing better because it resolves the ECC junctions on the basis of local de novo assembly, which is lacking in Coral and Decoy. So in the next part of my talk, I would talk about the microDNAs and how we are thinking that we can use them as biomarkers for early stage breast cancer detection. These microDNAs are typically less than 2000 base pair in length. They produce microRNAs that can regulate the gene expression. They can be detected in both normal and cancer tissues with the potential of being used as a biomarker. In the figure, I am showing an electron microscopy figure where we could see that these microDNAs are shown as double-stranded and single-stranded as well. So coming to the methodology, for the library preparation step, we start with the cell lysate. After extracting the DNA, we digest the linear and mitochondrial DNA, only keeping the circles intact, which we then rolling circle amplify, following with the library preparation step and sequencing. I would like to thank Dr. Kendall Walton for making these libraries.
So a quick question, Sarmad.
Yeah,
How
yeah.
can you guarantee or make sure the digested DNA are circular ones? By what protocol or something that you select?
Undigested DNA is the circular one or digested?
Yeah, digested, the digested one.
We don't digest
No,
the
not yeah, you digest the linear DNA, right?
linear and mitochondrial DNA, yeah.
Okay. So after the digestion, only the circular ones are left, right?
Yeah, yeah.
Okay.
Mhm.
So there's a kit or something that can achieve this, right?
Yes, there is like enzymatic digestion, which are like for the mitochondrial DNA, I think we digest it with PAC1 enzyme, which is having a restriction site. And for linear DNA, we digest it with the endonucleases, which chop off the linear DNA from 5' to 3' end. Thus, the circles which are not having the restriction sites and not having the ends exposed remain intact, and then it could be then subjected to the library preparation.
Okay, thanks.
Mm-hmm. So these microDNAs arise from the linear DNAs, and the difference exists at the junction sequence. After the rolling circle amplification, we identify these circles on the basis of the split read, which is a split read in the paired end read, which maps on the junction tag, which is a part of the discordant pair read. Discordant pair reads are the rows read pairs which are mapping on the position, location, and orientation which are different than expected. So on this basis, we identify the circles. This algorithm using the short read sequencing was developed by Dr. Pankaj Kumar when he was working in the lab. I would like to thank him for providing this slide as well. So we had like 25 paired blood plasma libraries for stage 2 and DCIS breast cancer patients. And we have analyzed 12 of them, and we are in the process of analyzing others using the rolling circle amplification, followed by analysis with circle finder. So after the analysis of microDNAs using the rolling circle libraries, we wanted to know that whether these microDNAs are coming from the overamplified regions in the cancers or not. So for that, we used the database ECGA. This database has a gene level amplification data, and we checked our genes which are outputting these microDNAs so that they are present from these overamplified regions in the cancers or not. So coming to the results, my results parts are divided into two portions. In the first portion, I would be defining the similarities between the pre-surgery and the post-surgery set. So these are the characteristics length peaks of microDNAs. So they show these peaks at the length of 200 and 400. And we could see these characteristics present in the both pre-surgery samples and post-surgery samples as well. We wanted to see that whether there is any specific enrichment for the genomic sites in the pre-surgery or the post-surgery set, but we could not see any kind of specific enrichment, and all these microDNAs in the both of the sets show a similar sort of pattern for the genomic sites they arise from. So we also normalize the libraries for the reads mapping on the microDNA bodies, and we could not see that kind of a difference between the pre-surgery set and the post-surgery set. So coming to the different results in the pre-surgery and post-surgery, we did the PCA analysis for genes for microDNAs. And the PCA showed that microDNA from follow-up samples seems to come from common gene, whereas baseline come from various genes. We believe this is because microDNAs from the baseline came from a different set of genes in the breast cancers, which are removed post-surgery, so that now microDNAs originated from some same normal tissue, the normal genome. So the microDNAs arises from the 22 genes exclusively in the pre-surgery set, not present in the follow-up sample. We also tested this in an independent sample set from fibromyalgia samples, and the microDNAs were not present in that set as well. Absence of these microDNA in the follow-up sample hints that they were exclusively produced in tumor cells which are resected. So in our result, we saw that two genes KIAA1549L and CDC14A are present in 7 out of 12 breast cancer patients, and three genes FINE, ERB2, and POLR3A are present in 3 out of 12 breast cancer patients. So we noted that 17 genes from these 22 genes are previously reported to be amplified in breast cancers, whereas 5 genes are amplified in other cancers. So for now, we have analyzed 12 paired samples from baseline and follow-up. We say that the microDNAs are coming from the pre-surgery are different with respect to the genes. There are about 2,100 microDNAs which are uniquely mapping to these 22 genes. The 22 genes are reported to be over-amplified in publicly available whole genome sequencing data as per the ACGA database. So coming to the discussion, these 22 genes have some of the genes which are already been reported to be prognostic in breast cancer, like ERP2, RECQL5, PBX1, and ROBO1. Well, there have been studies that these microDNAs are typically enriched at the transcriptionally active sites in the genome from our lab as well and from other labs as well. But here we are finding that these amplified regions also produce more microDNAs, so which is very interesting for us. So coming to these selective microDNA productions from the amplified region can be used to detect cancers. The absence of these specific microDNA from 22 gene in fibromyalgia supports our hypothesis. We are trying to validate these findings in our sample set and other independent cohorts. So coming to the acknowledgement, I would like to thank Dr. Anindya Datta for his support and guidance. All the members from Datta Lab, especially Drs. Yoshiyuki Shibata and Tsuko Shibata, for their input. Kendall Walton, who made these breast cancer plasma libraries. Sabah Tufail, who helped me to validate these long circle finder prediction for PCR validating the ECC junctions. Dr. Divya Sahu, my fellow bioinformatician for bioinformatic insight. I would like to immensely thank Dr. Zichen Chong, my co-mentor. Without his help, I would have not been designed or a long circle finder and learned bioinformatics from a lot of bioinformatics I learned from him. I would like to thank Dr. Yanfeng Zhang for his input. I would like to thank research computing team Chiha at UAB and Dr. Wendy DeMark Weinfried for the collaboration for the breast cancer plasma samples. And I would like to thank Dr. Colleen Jackson Cook from VCU for providing the sample set for the fibromyalgia sample set for the validation. Yep, that's it.
Thanks for the great presentation. Anybody, if you have any questions or comments, feel free to unmute yourself or enter your questions in the chat. I just have a quick question to get things started. Why is fibromyalgia plasma selected to be as a control? You know, there are other, that seems like it might introduce more variables and there's other markers of fibromyalgia. What is the reason for that?
Actually, we wanted to test these over-amplified regions in the fibromyalgia set, and there is no reason that the microDNA should be coming from these fibromyalgia sample sets. So we just wanted to test the hypothesis on an independent cohort, and luckily we had a plasma sample set in our lab, which were supposed to be fibromyalgia samples. But we are looking to check these hypotheses in the normal samples as well. You are right.
And what are next steps? Like you're starting out here with a smaller sample. Are there larger data sets available with similar sequences or plans to do any kind of larger?
Yeah, for the microDNA studies, we are having 10 more libraries which are being sent for the sequencing. We have already applied for the data access for the microDNAs from breast cancer tissues. So we are looking for the more samples to get the more power and statistical significance to our hypothesis.
Other questions or comments from the audience?
Yeah, I have a question regarding both the large circles and the smaller ones in terms of how they're generated and how they may be amplified. For example, with the larger ones, is it known how they do they replicate as autonomous plasmid or episomes, or are they
Well,
just constantly being spit out?
well, what we know is that they are like in missegregation and they are being replicated as well. The larger ones for that we know.
So is there any direct evidence of their replication structures or replication intermediates? There would be two basic models. One would be internal rolling circles, which I strongly doubt, and the other would be theta form replicons.
Mhm. Well, I need to check for that. Yeah, I
Because
cannot. Yeah,
because that would also presumably require specific replication origins.
yeah. But what I know from my knowledge is that they do have origin of replications, and like there have been some studies for that. But I need to check for that.
Okay.
Yeah.
And what about the smaller ones?
I don't think so that smaller ones are replicated independently. They are too small for that.
So they're presumably spit out somehow
Yeah,
through a recombination process, I would guess, or some kind of a D-loop formations.
yeah, yeah.
Okay, thank you.
Following up on that thought, I'm just thinking that teleologically, that it would make sense for a tumor cell to have those be plasmids that can self-replicate. That makes it more aggressive and faster replication. I wonder if, so since these are being detected in plasma, I wonder if these are, these are being expelled, or these are just breakdown products when those cells, when those cells die. I'm wondering,
Mhm.
what about these circular DNAs?
Yeah, for the micro DNAs, like their production has been supposed to be much more stochastic in the field, and people think that they are kind of an apoptotic product, but here we find that we can get some enrichment. So there is no reason for selection of like 400 base pair entity, but here we see that if there is a larger amplification product and then small bubbles are coming out of it, so we could attribute their smaller presence of the smaller microDNAs to the larger amplification. So that could be the reason they are spit out. So they could be used as a biomarker. That's what we think.
How readily assessed are these? So is this easy test to do for these patients to be able to find these DNAs, or is this a complicated testing procedure?
For the larger ones or for the smaller ones?
For either.
Yeah, for the larger ones, like once we have detected the ECC junctions, we could go for the PCR amplification because they would be amplified as a structure variation. And then we could see that the presence of junction is correlating with the tumor presence or the cancer recurrence or not. And for the smaller ones, they are like slightly more complicated because we have to prepare the library in which the circles are being enriched and rolling circle amplified. And then they are subjected to the paired and discordant read mapping for the analysis.
Any final comments, questions? Well, if not, then I have Dr. Mehmood's contact info if you have any questions subsequent to this talk. And thank you, everybody, for attending a great presentation. Thank you for giving such a great presentation, and all the people that you've acknowledged and contributed to your work. Wishing everybody a happy new year and a great weekend. Thanks, everybody.
Thank you.